So, good morning, welcome everybody to the first session, and today I'm going to talk about cannabis in cancer again. We have heard very excellent lecture, actually very interesting lecture from Professor Velasco yesterday, and he gave a good introduction, so I can skip some introductory words, and I would really today like to stress the importance of the mechanisms that are appearing in the interaction of cannabinoids with tumor cells, and especially point on the problems that are preventing the translation from basic research to clinical application, but not for social or any political reason, but simply because we do not understand science, or we do, and it's very complex. So let me give you this menu. I would like to talk mostly about brain tumors, about glioblastoma, but also, if time allows, a little bit on breast carcinoma at the end, and of course show you our platform that would allow for a prediction of the response of the patient, so in the so-called individualized cancer therapy. As we heard yesterday already from Professor Neubauer, there were basically known two strains of cannabis. These are Cannabis sativa and Cannabis indica, which differ not only in the shape of the plant, but mostly in the amount, the levels of THC, and of course also the ratio of THC versus CBD, cannabidiol. In these are two major cannabinoids that are supposed to be most effective, besides also other components which have not been so far investigated to a great extent. However, he also pointed out that nowadays you can have on the market very, very different kinds of strains. Why? Because the breeders really plant the different species, different hybrids, to increase THC because of the market demands, of course, psychoactive effects. And so we have a variety of plants, of strains, and they give very exciting names to sell them better, which means that different strains are on the market with very different composition of cannabinoids, and the difference between THC and CBD is not so much evident, or actually a lot of them have quite a similar amount of THC and CBD out of these strains. But how to deal with this in clinics, how to deal with this when we treat patients? So the renaissance of the medical cannabis cannabis was in Israel and initiated by Dr. Meshula, and really based on his activities and promotion of the cannabis in clinical application, they were starting to grow these different strains of cannabis plant in the cultivars, in special greenhouses, and so we call them chemovars. In these chemovars, you can really define what kind of strains you want to grow, because if you put really great conditions, controlled conditions, so that you can get the same composition of the plant, the same amount of cannabinoids, or the same ratios in all the time. And so we have about 400 species of these different cannabinoids, but they grow practically very well defined, 124 strains, as shown here. An excellent paper by Professor David Meiri in Israel was published this year, and he showed that he analyzed all these 124 strains and here are these strains listed in this direction. These columns are strains. And here you have different cannabinoids, and he just measured all the cannabinoids, and he noticed that there are clusters. They can be clustered into the group of the strains that have high level of CBG, or they have more expressed level of CBD, CBD-A type, THC, or THC-A type. And so you can see that actually we can really define four more defined strains, of course with different amounts of other cannabinoids. Yeah. Other. Okay, that's fine. Yeah, this is not good. Okay, so now we'll hear better. So I hope you got the message from this slide that really you can have different strains, but they can be clustered into 5 groups. And then he identified these strains based on the pathophysiological role that they are playing, or at least a physiological role. What kind of effects do they have? And you can see here they were defined by how they impair the memory, how they make you happy or excited, what is the effects on the fatigue or depression and so on. And you can see that they are very different. Although there is a same pattern of the effects, of physiological effects, still you can have great differences. For example, right in sleepiness, the effects on sleep, and you can see that different strains have different strength of how they help you to sleep. So why is this? This is because they bind to different receptors in the brain. Why are these effects so well known? Of course, you all know about them, or basically the researchers found that in the brain you have different areas where these receptors for particular cannabinoids, especially now here THC was well studied and defined, are located. So there are different areas. I'm not going into details of this anatomy, but they are different centers for memory, for eating habits, for appetite, for example, for memory, for motor coordination, so movement, and so on. And also the CBD has different effects in the brain, although CBD receptors are more scarce in the brain. So you can imagine now that the pathophysiological effects also of the cannabis plant, as people know it, can significantly differ. Depends on the source of the plant and maybe even predictable. So this is the problem number 1, because people or even doctors are not sure what kind of plant to prescribe. And that's why it's now a tendency that we can purify from plant or make synthetic cannabinoids. And quite differences were found. Extracts from whole plant were more effective. Maybe also due to other components that are present there in minor amounts, and this is what they call entourage effect. So the effect of other components which are not well defined, and it's very hard to analyze each particular plant that you are going to eat or to consume. Also, we have to know that there are endocannabinoids. You probably know about them, and these are mainly these are secreted from different types of cells, especially in the brain. And these bind to the cannabinoid receptors. So the receptors are important. And as you can see here, receptors are really a big protein molecules that have different spatial configuration. And because of that, they can bind. They are flexible and bind this rigid kind of structure of organic molecules, and these molecules represent one or the other type of cannabinoid. Now, the way they act actually is pretty simple biochemical interaction between a receptor and between this molecule, and these bindings, as you can see here, receptor and endocannabinoids in this case, occurs between the two cells. Here are shown the two neurons. This is an example of how cells communicate. Also other cells, not only neurons. Neurons in particular communicate via synapses, and they secrete, one cell secretes neurotransmitters, the other one is accepting the neurotransmitter, accepting by binding on the cell surface receptors. They go into cell, and this causes some electrochemical signals, which triggers the production and secretion of endocannabinoids. So this is the way how cells talk to each other. And of course, this is going on and on all the time in our brains. The receptors, the major receptors for THC is CB1, and this CB1 is really, as you have seen before, present in brain in various locations, and the other one is CB2, which is distributed more evenly in the body, particularly taking care of our immune system, our immune reactions. Well, this is looks simple, but actually it's not that, because it's not only CBD and CB1 and CB2. There are other receptors present. So there are also other types of receptors. I'm not going into the chemistry or definition of these receptors, but as you can see, different cannabinoids, CBG, CBN, that I have shown before, can bind to different receptors. This is a very promiscuous interaction, actually. One receptor can bind several cannabinoids, and one cannabinoid can bind to different receptors. Of course, the differences in these bindings is the strength of the binding and what these molecules do to the receptor, how it's conformed, and this all affects the signals, the consequences. What are the consequences of this binding? The consequences are that here, when this binding occurs on the cell surface, and this is shown, this is a cell membrane, symbolically shown like here, receptors inserted in the membrane, but this is all flexible. This is not a rigid, like a key and the lock. It actually very flexible, so the receptors can travel along the membrane or along the cell, and of course then they catch the endocannabinoids that are around, and then the signal, and here are various molecules. I'm again not going into details, but there are several cascade of reaction, chemical interactions, until this signal reaches the cell nucleus, and as we all know, there is a DNA molecule. This is a molecule that is really directing the production of our enzymes and everything, and our genes, of course, and then genes produce this enzyme, and these all affect cell, how the cell behave. Either it grows, or either it die, or it migrates, or whatever, and this is all very important also for the cancer cells. Now, this slide reminds me that I have to switch to cancer finally, and tell you that the effect of cannabinoids, of cannabis, was known for a long time in history, as you all know, but maybe you don't know that it was also described by Hippocrates and his scripts, and Hippocrates actually gave the name to disease, cancer, means cancer, and it's what's called carcinoma. What was at that time revolutionary was that he said that this is a natural balance between different substances and our body, and so he also described plant extracts, including cannabis. So it has some basis, very empirical, of course, and now we know more about everything, also about cancer. I'm showing here a pathway how cancer develops. Here it's shown colon carcinoma, for example. This is a healthy tissue of colon carcinoma. You can see these polyps here, and these are like a normal polyp, and then a tumor cell appears, and for whatever reasons, especially due to the disbalance, again balance between oncogenes and tumor suppressor genes, to different molecules, types of molecules, and affect this cancer progression, as we say. And so the cells grows, and finally cells cannot stay on one place anymore, and they dissolve this membrane, or we call it basement membrane, around each organ, and they can metastasize all over the body. Now, growth, invasion, metastasis, these are the key events. But as we also heard yesterday already from Professor Velasco, is that you have many so-called hallmarks of cancer. So the typical properties, we call them hallmarks. And let's say we all know growth, we all know there's invasion and metastasis. We also know that there is special interaction with immune system. Immune therapy now is very much these days. They are immortal, they reduce growth of new vessels, and so on. And of course, they are resistant to apoptosis. They don't want to die, they just live forever. And now we have got through this publication. This publication again is from David Mayuri, and they have really used this receptor, they have used these cannabinoids to different cell types, and these are cell types, tumor cell lines from different types of cancer, breast, prostate, glioma, and so on, that have different names. And then he first measured how are the cells doing regarding the receptors. Regarding these different types of receptor that I have shown you before, and there are really numerous, and you can see that each type of cell can be differently expressing the receptor. So it's not only different cannabinoids, it's also different receptors, and this certainly is the problem number 2. So different types of tumor have different receptors. Even more so, the same type of tumor, for example, breast carcinoma, and this here is shown different carcinoma strains. For example, these strains, like 10, he took 10 strains, and then he measured the effect on the cell death. So some strains cause cell death. This is high, then this level is high, and some do not cause anything. And so in breast, we have seen that 2 different types of cells, of breast cells, still breast cancer, but different types, genetically different breast cancer cells, can respond very differently. This one is not responding to any of the cannabinoids, and this one is really increasing the cell death of various strains that were used. Now, for glioblastoma, it's about the same, so even same tumor can be very, very different, and this, of course, is the problem number 3. Namely, the same type of tumor would not respond the same. And we know, you probably all know, that you can have, in breast cancer, there are 12 or even more type and subtypes. In glioblastoma, there are 4 different subtypes, as I will show you in a minute. So glioblastoma, this is really a very lethal disease with only 1.5 year of survival, whatever you do, and its significant property is growth and invasion. And you can see here how these cells grow differently, maybe from carcinoma, as I've shown you before, that grows in a mass. Here, the tumor cells just spread all over the brain, even to another hemisphere, and that's why it's really hard to catch them by surgery, radiation, or even chemotherapy is only increasing on average the survival rate for 2 months. Now, which are the cells? So these are differentiated cells, but these cells are also very heterogeneous. That's another problem. You can have cells that we call tumor or cancer stem cells. And in glioblastoma, this is a minor fraction of the cells. If this is tumor in the brain, and there are different tumor cells here, and here you can, one cell type is different, have all the properties, all the program to develop the tumor, genetic program to develop the tumor, like, for example, embryonic cells have a genetic program how to develop the human body. Now here it's the same. All the information is in these cells, and of course we would like to kill these cells, but it's very resistant to all types of therapy. And we have shown that also this cell line express, for example, CB1 receptors. I will tell you more about that. Just to point out again that if you, let's say, reduce the volume of the brain tumor by any means, and then you can kill all these cells, other tumor cells, but these stem cells would remain, and would remain probably the same type place, or go wander a little bit around, and you will get a relapse very soon. So that's why we need to kill these particular cells. Problem number 4, but then we have also problem number 5. Problem number 5 is that these tumors, as I said to you before, there are 4 subtypes, and these subtypes are genetically a bit different, so they would also perhaps express genetically programmed different receptors, which would differently respond to different cannabinoids. Great variability, but even one tumor can have these subtypes, which we call classical, neural, pleural, mesenchyma, and so on. And all these cells, all these 4 types of cells can be present in the one tumor. then how would you distinguish? Impossible. So it has been shown that if the tumor is very heterogeneous in this respect, you get the reduced survival rate. This was shown mainly in animals, but also in autopsies, analysis of the composition of the cells in the tumor, and the patients have different survival rate. The mesenchymal type is really most dangerous, most aggressive, and have shorter survival rate. And even more so when tumor relapses, it kind of transformed into a mesenchymal subtype. So this is a problem. And how to approach now the therapy with cannabinoids, knowing all these obstacles. Now we have tried to, in our lab, we have tried to analyze the patient's tissue, the patient's tumor cells, what is in the tumor, what is in the patient's tumor, and we get from the neurosurgery in Ljubljani University Clinic department, we get the tumor pieces at operations, and we cut it in pieces, and from this there is a protocol how to grow primary differentiated glioblastoma cells and how to grow glioblastoma stem cells. These are grown always in spheroid, in three-dimensional form, whereas these cells are more adherent differentiated cells. But anyway, we then collect these slices, and also this tumor is cut into tissue sections, so to say, and this is done at the Department of Pathology of Medical Faculty, and these sections are used for genetic analysis. So we would like to genetically analyze what kind of subtype is there and what kind of receptors are present in a particular cell, stem cells of the particular patient. As I said before, these stem cells here growing also express CB1. This is a green color, CB1 receptors. Also express CB2 receptors, and these are all established cell lines, and also cell lines for patients would behave the same. And when we staining means that you have a antibody against these receptors, and this antibody reacts with the receptor on the cell surface, and when it reacts, it gives a certain color, which we call immunolabeling. So the color, and here this brown color are all receptors, CB1 expression in tumor tissue, so that's how a section of a tumor tissue would look. And here another patient, and then CB2 is also expressed, CB1 and CB2. But when we isolate the cells from the same patient, we can also see that also these cells express CB1 and CB2. I have to stress here that tumor cells express more generally known, more receptors than the normal cells, whereas maybe also other cells that are mixed in the tumor, like macrophages infiltrating lymphocytes and so on, can also express these receptors, but not to such a great extent. And that's why this receptor can be the target for the therapy, for the cannabinoid therapy. Now here are just examples of different patients. We have collected cells and we have collected tissues, so to have a larger statistical data to correlate them with genetics of this patient, of this particular tumor. We have made some analysis also of this receptors expression, for example, of CB1 receptor in different stages of malignancy of the glioma. And as you know, this is glioblastoma. Glioma can have different stages, less aggressive, less malignant to highly malignant glioblastoma. Glioblastoma is the most malignant stage of glioma. And here you can see that we really found a lot more of this receptor, as I told you before, correlates with malignancy, although we don't have enough data yet to see if glioblastoma stem cell would have more or less than the differentiated glioblastoma cells. But in any case, we are collecting this data now. It's a process of our study, so we need more data to get better statistics out of this. Now, finally, of course, we take the tissue and we test the effects of cannabinoids on the live cells of the patient's cells, live patient cells isolated from this tumor. And so we have seen the response, we measure the response to THC or CBD. Here, this data show we have used an enriched resin. So this was not purified, this was a resin that you can also get from some producers, and people are also using it for mainly self-remedy. So here we have seen the differentiated cells and the stem cells below. And we have perhaps seen that THC may really be more efficient for glioblastoma stem cells, which is a great news because, as I told you before, these cells are the most resistant to all the therapies. Now, here it looks like that. It looks like CBD doesn't do much. To neither of the two, and then we put them together and change the ratio between the two, and we have seen that it's kind of synergistic effect in glioblastoma line as well as in glioblastoma stem cell line. Same was seen in the patient, Same was seen in one patient, for example. That's just one example. When we have mixed THC alone, it is effective. CBD alone is effective. But when we mix the two in 1:3 ratio, for example, it was we have seen the reduction of the tumor cell viability. So its cytotoxic effect was increased when the two were mixed together. And so we did then try something else because we don't know what actually is all in this resin. And we started very fruitful collaboration with MTC Pharma, pharmaceutical company that is also located in Ljubljana, has actually a headquarter in Ljubljana, and they are producing purified, but natural, not synthetic cannabinoids, purified THC and CBD, and we have then used them again to test the same cells. Now we have observed differences. Here, when purified, CBD was more effective on glioblastoma and glioblastoma stem cells compared to THC. Actually, we had to use more higher concentrations of the purified stuff. So we have determined what is so-called IC50 values. These are the values that reduce the viability, or in other words, induce that are cytotoxic so that they kill 50% of cells in our culture. So we have seen, in this IC50, the lower it is, the more toxic is substance, and we have seen that there is a difference actually between the differentiated and non-differentiated cells, stem cells, and but the differences are not very significant. Actually, we don't know because we have to test. We have to test. No, sorry. This is the right one. We have to test more patient cells to see if there's true really in vivo, in tumor patients, if the stem cells of the tumor patients are more resistant or less resistant, or actually we want to know what in combination can we use, because this is only a single effect of CBD or THC. Now then we will try a different combination, hoping to get some synergy at some particular ratio. In clinics you have seen, you have heard from Professor Velasco yesterday that they are mostly using preparations that are about 1-to-1 ratio of THC and CBD in the clinics. However, we don't know if this is really optimum for a certain patient. So this is our plan now. This is just a method how to measure. Why is the viability reduced? Because cells die more often and do not proliferate. I'm not going into details of this experiment. Also, what we do is to study the signaling effect. This is perhaps too complicated for some, so you cannot skip listening to this explanation, but we have, but this slide, for example, shows you this complex signaling interaction in the cells that really cause all the changes in the nucleus, cell death, cell survival, and so on, when you put CBD or THC or combinations to the cell. And this signaling is affected so that CBD would prevent the signaling. So all these molecules that are in the cascade that are excited when the signal comes are not so excited when you add CBD. And this is shown in this slide. We have used two different types of differentiated cells. U87, this is the standard, as you have already seen, U87, U373, these are the names of the cells that people are using most. That's why we are using this as a control or as an example. And here we have put CBD. You can see that these cells would respond, the change in the shape is quite obvious. Then we have used also temozolomide, and this is a final goal, that we would do the combination with temozolomide and cannabinoids, as you have also heard yesterday of, that this is where the clinics is going. Clinics will never use only cannabinoids. In clinical practice, we will use a combinatorial principle. So we have used here temozolomide. Indeed, the cells are even more disorganized, disrupted, and here we have used the two, and this is really highly effective in causing cell death, but this cell line did not change so significantly. So, and also viability of this cell line is not affected. So with U87, yes, and then we have looked at what happens with this, one of the signaling molecules called ERK, and ERK is really downregulated in these cells where cannabinoid was effective, and not at all in this cell that cannabinoid was not effective in reducing the viability. So this is kind of experiments that we are using just for basic research, not for the clinical application. And to summarize this part, this was published also last year by Dimitri, a review what all cannabinoids do to glioblastoma cells. They affect invasion, they affect tumor growth, and they affect glioblastoma stem cells. So this is a very important news, again, because glioblastoma stem cells are affected by cannabinoids, not by other therapeutics. And this is a way to go future and see what specifically would affect them. Now, for the practical preclinical data, we have constructed, or we are constructing the predictive platform, which means that we are doing these tests that I have shown you before on the fresh tumor tissues and fresh tumor cells to then characterize what subtypes is, how is the level of the expression of cannabinoids, and how this particular cannabinoid or combination or whatever, and in combination with therapy, would affect the cell viability, toxicity, and so on. And so then, of course, there's a lot of data. We are grinding up this data by bioinformatics tools and algorithms, and then we can hopefully come to a decision that if the patient has this subtype, this level of receptors, it would probably respond best to a certain combination. And this can be done in vitro on the tumor of patients while the patient is waiting for the therapy. And when we get this information, we can go to the clinicians and say, well, this is in vitro. It doesn't mean that it would really be so in the system, in the body, but it's high probability that it will really be. So we hope that this will be a preclinical platform that can be established in many labs, actually, although you need to have a certain equipment and knowledge how to do that. So, and then we hope to improve the clinical trials. I will not go into details of this because it was already mentioned or discussed extensively yesterday, but the summary of the clinical studies or the animal studies is that in glioblastoma, this was demonstrated that cannabinoids cause significant reduction of tumor volume and sometimes even complete eradication. This was published in this review by Drs. Licker and Naller from Klagenfurt, and they also have shown that in animal experiments, again, concomitant treatment with X-ray or temozolomide did enhance the activity even further, so extend survival rate of the animals, and the combination also cause synergistic effects, and this is what I have shown in our cell lines already. So at the end, I'm not repeating what we all know, that it's approved also in our country for palliative medicine if the doctor wants to use it or if it's discussed among doctor and the patients. But of course, all this we know, no side effects, which is very good, but it's still not approved that we will, during this, that we will administer, or the doctors will administer these cannabinoids at the beginning, at the diagnosis, and follow the reduction of the tumor, or tumor regression and so on, this, or measure invasion and so on. This is not in a clinical use, and it will be probably sometime. And of course, the problems, I listed all the problems. There are problems in the different level of receptor, different strains of cannabinoids, different compositions of cannabinoids, different ratios, genetic subtypes of the cancer stem cells resistance, and of course synergy with other treatments. I have not mentioned that, but very few drugs have been investigated so far to not lose their effect when you add cannabinoids, because this is a fear of oncologists, that if they would administer cannabinoids, they would lose the effect of the standard treatment. And this has not been shown, as I showed you, for glioblastoma, also for breast cancer, and for tamoxifen and certain therapies. But it has been shown for some other drugs. And this is an area, still a gray area in our knowledge, and we have to investigate it further. So again, we need to solve all these problems. So what we to do? More translational research and more basic research. And of course, maybe we can really test the cannabinoids in a better in vitro models, like in organoids, perhaps. So we are already using three-dimensional structures to test, because these are more similar than two-dimensional monolayers of the cells that people are used to use for the drug testing. So drug testing, which is not only for cannabinoids, but a general in pharmaceutical industry, a problem. And then, of course, also better animal models. Mice are fine, but they're very expensive, and we are now trying to do the zebrafish, which is quite effective model, quite comparable model, which is much cheaper and faster. And of course, clinical studies all need to be personalized. So as this lecture showed you, each patient can be very different and can respond very differently. So course take-home message is that patients that are seeking for cure, not only for relief or their pains or problems, but to increase the life quality, that's fine. But if you want to really cure the cancer, you should use well-defined and formulated preparation prescribed by a medical doctor. That's, at least is my opinion, and that would be tailored to individual patients' profile and of course, the needs. So I would finish with that and thank my collaborators or my team in the Department of Genetic Toxicology and Cancer Biology, where we have this cancer research group, mostly girls, as you see. But there's a man, a collaborator, Professor David Meiri, that I presented some of his very recent results, and we think that we will collaborate along with his cannabinoids extracts that are well defined. And of course, there are also some other, especially our neurosurgery clinic, and so our partners in a bilateral INTERRIC program, transglioma, which is really actually providing or helping us to build the so-called biobank, tumor bank, and this we do in 2 countries. So at last, I would like to thank you for the attention, and I think that this strain of cannabis is effective, and it's not an utopia of the future treatment. Thank you.
Tako, hvala vam. Ima morda kdo v publiki kakršnokoli vprašanje v zvezi s predavanji gospe Tamare? Ne?
Not yet.
Dobro.
Maybe by the time.
Ja, 10 minut rabimo, ko Imbal pride. Vsajno malo prav.
Dobro, 10 minut prostora se je zgodilo.
Well, maybe I can tell you something about breast cancer.
Please.
If there's time, so I was really pressed by time. But still, breast cancer, well, this is what I was telling you before, that cannabinoids can be affected by other drugs, and this is problem number 7, if you want to count them. And here, what has already been shown in the literature is that cannabinoids, CBD, for example, is synergistic or not, but it can be combined with different types of therapy that are using for glioblastoma. These are chemical or small drugs, small molecules. And also in breast cancer, we are now investigating tamoxifen, which people have also been wondering if it is compatible with cannabinoids because the receptors are similar and can interact. But we have found just the other way around. And so this is also, of course, important for other drugs, and for combination of drugs is always a problem, as you know from your everyday life, perhaps. Now, breast cancer is now also even more serious problems, on one hand because it's not a rare disease like glioblastoma, but the breast cancer is a more spread disease. It's very frequent, actually it's the most abundant, highest incidence in women. And a lot of investigations have shown, of course, the treatments, you can see different kinds of treatment that you can then use, or the diagnosis first and then treatment, have been, especially mammography and screening, has increased the survival rate significantly, and so it's not anymore a deadly disease like glioblastoma, but it's becoming a chronic disease. Of course, a cancer patient is practically never cured. It has to follow up all the time to really be careful on the appearing metastasis, even years after the primary tumor is operated. So you can see here different types of diagnosis, procedures, and so on. And again, here there are subtypes. Subtypes of breast cancer can be like hormone positive, so they have still the estrogen receptors, the progesterone receptors, which are the receptors that are normally functioning in the breast, regulating the activity of the breast, especially during pregnancy. And this systemic therapy has also been improved. What is as pre-operative, so neoadjuvant, so you don't cut out the tissue, you do different approaches, also biological therapy and so on, hormone therapy. Then it's post-operative, which is adjuvant to this, or irradiation and so on, and it should be then administered to a certain extent, and the oncologists are pretty well aware of this. For this study that we are using in doing in collaboration with the Institute of Oncology in Ljubljani, we have selected specific established breast cancer cell lines that you can get in the cancer cell banks or so on, and then you grow them, and what is really specific for these cells is that they represent different subtypes of the breast tumor. As you can already see here, they have different morphologies, so they certainly are not the same cells. These, for example, are obtained from breast metastasis, still breast cancer cells, but already at the metastatic site. And so we have the following workflow, what we do investigate. So these cell lines, as you can see here, are all, of course, from are not from patients, but are from banks, but they are originally, of course, isolated from patients and established as a stable cell line. And then you can have a subtype, which is called luminal A, B, basal, and so on, and then one have receptors of some growth factor, which is called epidermal growth factor, and the or HER9 gene, and this can respond to the antibodies to this receptor, which we call, it's called Herceptin. So you maybe heard that in treatment with this biological therapy is very effective in women who have this expression, but not all women have it. Also, not all women have receptor positive, so status is very different for these subtypes. Okay, so now we have then used these cell lines to test first the amount of the receptor CB1 and CB2, and then of course, and also do all the immunochemistry of these receptors, and also then try how they respond to cannabinoids. So these are the so-called Western blot. You can see the level of receptor protein here, CB1, CB2. Most of the cells, but not all, express CB1, but most of the cells express CB2 receptor. Not going into details, I just want to show you the kind of summary experiments that we made so far. We have taken CBD and take this cell line, T47D. This cell line responded well to CBD, as you can see here, up to 50 micromolar, and then we have added tamoxifen. Tamoxifen was not so much effective, but when we add tamoxifen and CBD together, we got a good response to tamoxifen. Better response. Just to finish this slide, I will be in a minute. But now there's another one, and here we have used not cisplatin, the estrogen antibody, and here we have also used that this does not respond by itself, but it responds better when it's CBD is added, and here CBD also affects the MDA cells, but when you use cisplatin, which is another chemotherapeutic, this is synergy. So we have great hopes for such a therapy also in breast cancer, combinatorial therapy, and this research is ongoing. Thank you for additional